I'm going to talk about a line of research that looks at how our social environment essentially regulates the activity of the human genome. And I realized right as I was putting the slide up here that I hadn't converted the human genome to primate genome to create a greater apparent articulation with what might be going on here. But nonetheless, you'll see there's monkey data in here that suggests that what we're seeing in the studies that we predominantly focus on in human beings is probably part of some deeper, I won't hazard a guess about how broadly conserved it is, but some sort of programming that allows the human genome at the very least, probably primate genomes in general, to a little bit tailor their responses to, if you will, sort of the cellular or biochemical world based upon the social world surrounding us. That there seems to be buried in the regulatory logic of the primate genome some distinction between the sorts of genes you should express when you're around others of your own species, especially if they like you, and the sorts of genes that you should express if you're not, if they hate you, or if there's no other conspecifics around. But what I'm going to do at the beginning is actually back up a little bit and talk just a bit about a sort of a surprising discontinuity between the way we experience our bodies and the way that we know, or at least are increasingly beginning to appreciate, how they actually work at the molecular level. So we generally experience our bodies as stable biological entities that live in the world but are separate from it. In other words, I feel like I'm the same person today as I was a couple months ago, and I feel like, at least at the corporal level, I am the same person if I'm in this social context as if I were in a completely different social context. I am me. I'm kind of this rock-solid invariant wandering around through the world. When we look, though, at the biological underpinnings of me and other representative primates, we find that we're actually far more fluid at the molecular level and far more permeable to environmental influence than our intuitions would typically have it. The fluidity comes from the fact that basically we're constantly involved in a process of self-regeneration. So the average protein in the human body has a half-life of about 80 days. That means every day I need to replace about 1 to 2% of the molecular me. And that sort of regeneration or replacement process is mediated by the ongoing regulated expression of genes. So stretches of DNA that include the so-called coding region of the gene, the part that's actually going to be translated into protein, basically, as well as upstream sequences that control whether that coding sequence is actually expressed, whether it's actually transcribed into RNA and translated into protein later. So if a cell is basically, essentially received the message that it needs to make more of a certain protein for one reason or another, it will activate a transcription factor, a protein that binds to the regulatory region of that DNA stretch and flags the coding region for active transcription. In the absence of that kind of an affirmative signal, this gene will sit there at the DNA level having no influence on your molecular biology at all. It'll be essentially a silent influence. It's a potential influence, but not necessarily a realized one. And as we've developed the capacity to measure the expression of genes at a massively parallel level, so we can easily now poll the expression of all 22,000 or so human genes in any particular sample of tissue we want to look at, what we've discovered is that this process of sort of regulated transcription of our genomic potential into an actual RNA transcript that can then go influence our phenotypes is actually more an exception than the rule. There's a great deal more encoded information in our genome than is actually actively expressed in any particular cell type. So there's quite a lot of, if you will, contingency or decision-making, regulatory, sort of instruction going on when we take our genomic potential and actually have it realized in the context of any particular cell. The majority of human genes actually aren't expressed in any given cell type. Most cell types express only a minority of genes. So there's quite a lot of this kind of contingent decision-making going on. So that's the, if you will, the fluidity of this process. It's not like my molecules have been with me since the day I was born. Lots of, sort of physical components may have been with me, but some of these key things like the proteins that are actually making cells and bones and the rest of us do the work that they do, that has to continually be replenished, and it's replenished in a way that involves a certain amount of decision making. So the fluidity comes from the fact that we're constantly, in some sense, recycling ourselves. The permeability comes from the fact that this regulated recycling process basically is taking place within a cell that is essentially somehow cognizant of the outside world. These cells bear receptors that allow things like the social environment, as converted by our brains into biochemical representations, to influence this regulated gene expression trajectory. In other words, to the extent that we, for instance, feel threatened or overwhelmed, that has these knowable biochemical representations that then interact with receptors on the surface of the cell, and it activates these biochemical signaling pathways that eventuate in the activation of these transcription factors. These receptors also cause lots of other changes in the cell, but for gene regulation, what's particularly important is the fact that these receptors that provide cellular, if you will, awareness of what's happening outside the cell and potentially even outside the body, that has the potential to hook up to the activity of at least some of these transcription factors and control which subset of genes is being expressed, and by complement, which subset of genes is not being expressed in a particular cell. So this creates this, in principle, possibility that the social environment can change the way our genes are actually, if you will, behaving, the pattern of expression of these genes. And what we do in our lab is we basically try and understand the empirical realization of that conceptual possibility. In other words, we're interested in determining which types of genes are subject to socio-environmental regulation, which transcription control pathways carry social information to the genome, and a little bit about how genetic polymorphisms might render some individuals' genomes more or less sensitive to this kind of exogenous socio-environmental regulation. So one way that we go about that is to, as I mentioned earlier, apply the sort of actually spectacular developments in our ability to measure RNA and DNA in this massively parallel way. So we can look at all 22,000 or so genes in the human genome, or in this particular case, we're doing it in the rat genome. And then we can do all kinds of studies, either experimental manipulations of social conditions, or we can do observational studies of humans as they confront sort of challenges or opportunities in what you might call sort of a free-range human life, and see which particular genes get activated, sort of systematically as a consequence of that, and which are left untouched, and which seem to actually be downregulated. So here I'm showing you results from a representative study where we take essentially adolescent rats that have just been weaned, they're capable of living on their own, and we randomize them either to continue living life the way rats typically do, at least in our labs, which is in a big pile of other rats, with all kinds of social stimuli and a certain amount of maternal, sort of structuring of these kinds of social dynamics, or they're randomized to spend another couple of weeks in isolation. It's not painful isolation. It's a perfectly comfortable little rat apartment, nothing particularly aversive about it, but you don't get that kind of social stimulation. And then after about, maybe I think this case we did about 3 to 4 weeks of isolation, we can look at any particular tissue in the rat body that we might care to and ask which genes empirically seem to be upregulated as a function of this experience, which genes seem to be downregulated, and which, by complement from those two sets, aren't appreciably regulated at all. So what you're seeing here is what's called a heat plot representation of about 1,800 genes that showed greater than 50% change in average expression level in isolated animals relative to the control group that was socially housed. In a heat plot, the rows correspond to individual animals, the columns correspond to particular genes that are in this subset that was found to show greater than 50% difference in average expression level, and the extent to which the cell defined by their intersection is red indicates the extent to which that gene is upregulated in that individual. The extent to which it's colored green indicates downregulation. If it's black or somewhere in the middle, that indicates not particularly substantial change in expression. So what you can see in these reasonably well organized blocks of red and green is about 1,500 genes that are notably upregulated in the isolated animals on the average across the, I guess, 5 or 6 animals that we were looking at here, about 300 genes that were selectively downregulated in those animals. Now this turns out to be about 9% of the total rat genome actually changing its expression level. These, I probably should have told you which particular tissue we're looking at. Some tissues you might imagine don't pay much attention to your social life. We haven't empirically looked at the liver, but I sort of imagine your liver is just busy doing liver things most of its life and doesn't really worry about this. I could be wrong about that. But certainly, the prefrontal cortex, which is what we're looking at here, does, it turns out, pay quite a lot of attention in terms of gene expression to these kinds of socio-environmental conditions. So on some level, this is not terrifically surprising, right? I mean, the brain is the fundamental organ not only of our perceptions of the social world, but a chief mediator of our behavioral responses to the social world. So it's not, you might think profoundly counterintuitive that the brain would be turning on genes and turning off genes in the prefrontal cortex as a function of these fairly profound differences in social stimulation. What's been more surprising to us, though, is the extent to which, as we survey other tissues of the body, we find that they're paying attention to the social world as well. In other words, information isn't just getting in our eyes, bouncing around in our brain, and then going back out our social behavior. This stuff is also, it turns out, trickling down into the rest of our bodies to change gene expression profiles down there. And that is actually sort of a little bit of a head scratcher. Why would we, want to hook up the fundamental cellular molecular biology of some of our most basic physiologic systems to this kind of supposedly transient fluctuating external socio-environmental stimulus? Why, for example, when we look at this study of circulating white blood cells, would we see a couple hundred genes that are sensitive to the experience of being socially isolated versus integrated in human beings? So these are data from a study where John Cacioppo has been following a cohort of about 200 older adults in the Chicago metropolitan area now for, I think, about 8 years. And maybe about 2 or 3 years into that process, we had an opportunity to sample white blood cells from those individuals and poll gene expression, and we found about 200 genes that were showing systematic differences in expression, about 70 relatively upregulated in these lonely, isolated individuals, about 130 relatively downregulated. So this is a little bit more of a head scratcher because, from a, if you will, sort of a biomedical model perspective, these immune cells ought to be running around paying attention to bacteria and viruses and the other kinds of things that they're there to manage, to combat. And it's not clear why they should be, in some sense, at the gene expression level, behaving themselves differently as a function of people's social environments. These people are all healthy, older adults. They're selected so they don't have any overt disease that we can, at least that we can detect. And so, there doesn't seem to be any reason that we would expect a priori these kinds of social variations to map onto changes in gene expression, at least at the level of the pathogens that the immune cells are, sort of, we believe, teleologically there for. Even more surprising to us is, as we've followed up that initial study of lonely older adults, by looking at other populations of humans, now even extending this into randomized social experiments involving monkeys, particularly social instability and social isolation, we have begun to find a surprising degree of consistency in the nature of the genes that are upregulated and downregulated in cells of the immune system, in individuals confronting a whole variety of what you might broadly call adverse social conditions. In fact, even a night of sleep loss is enough to change the expression of a few tens of these genes. Certainly not as profound as the hundreds of genes that we've seen, but relatively transient influences have, at least some sort of primordial or vestigial kind of play on the same kind of gene expression dynamic. And so we have spent a fair amount of time trying to map how these kinds of experiences come to be expressed in terms of changes in gene transcription. And we know, in principle, a fair amount from decades now of studies of what I'll call broadly social signal transduction, how the social world gets converted into changes in cell biology, and then within the last decade or two, our ability to actually translate those stories about the nervous system and hormones into changes in gene expression has, of course, expanded quite dramatically. But in principle, we've known about much of this system through a slow accumulation of insight for, actually at this point now, upwards of at least 50 to 100 years. We know that the social world basically can become represented within the body via the brain's ability to, in some sense, convert social information into, if you will, sort of computed projections about the significance of external conditions for individual well-being. In other words, we can experience a sense of threat, or we can experience a sense of security, we can experience senses of hope, optimism, the ability to acquire things that we care about, or we can have the opposite of that. These kinds of fundamental computations or translations of experience into what you might think of as sort of conserved psychological representations then have the capacity to activate what I'll call peripheral distribution systems, broadly speaking. These include the nervous system, principally the sympathetic nervous system, and the endocrine system, particularly the hypothalamic pituitary adrenal axis. Both of those systems are especially sensitive to experiences of threat or uncertainty. Although they actually tend to activate in slightly different ways in response to, if you will, somewhat not so subtle, but at least distinct types of experience threat, which I may end up talking a little bit about later. But in any case, our brain has the capacity to change our perceptions of the outside world into some kind of biochemical representation that then gets distributed throughout our body. The biochemistry of these social representations essentially then can interact with receptors on the surface of cells that can activate these transcription factors that are essentially hooked up to the receptors on these cells, and that in turn regulates the activity of our genome. So again, this is the in principle pathway. This is not to say that there's not other influences that come from people experiencing, difficult lives who decide to smoke and drink and do all kinds of self-injurious things. But even above and beyond what you might call this kind of physically or behaviorally mediated change in your tissue biology, there's this, if you will, psychologically mediated change that is in principle capable of occurring as well. We know in particular that this step right here, our experience of the world and our representation of it as essentially a good thing or a bad thing, a promising or a threatening scenario outside us, is a key event in the system. And we know this because when we look at the correlates of these kinds of socio-environmental variables in gene expression, we tend to find much larger and more robust differences in gene expression as a function of people's subjective experience of the social world than we do as a function of the objective characteristics of the social environment itself. So in this particular scenario, for instance, we can ask people how much, essentially how lonely they feel. To what extent do they feel like, if something bad happens to them, that there's people in the world that they can turn to for support, that there's, in particular, either specific individuals or that just, humanity in general is good, kind, and decent, versus people who feel like, actually I'm pretty isolated. It's really me against the world. If something bad happens, I'm not sure who I would turn to, and basically you can't trust humanity. People in general will rip you off. That turns out to correlate much better with differential gene expression than does the objective degree of social integration that we see in these individuals. In other words, if we go out and measure how many relationships they have, how many close ties or bonds they have, if we measure the number of people they talk to on an average day, that has a little bit of a claim on gene expression, but it's basically about an order of magnitude smaller than the claim that this, if you will, subjective social worldview has on gene expression. So we know that that CNS-mediated dynamic is important, and that's probably why so many different kinds of socio-environmental adversities, if you will, end up having such similar changes in gene expression associated with them. It's because probably our brain at some level is converting them all into some similarly computed view of whether everything is fine and I've got great opportunities and the world is a great supportive, affording place, or it's actually an uncertain or threatening and hostile place. To the extent that we're getting these big sort of threat experiences, you can imagine how all of these different kinds of external social conditions might end up playing out in similar ways as it cascades down this pathway of social signal transduction. Okay, so like I said, sort of in graphical terms, what we try and figure out is which genes are hooked up to this social signal transduction pathways, what those pathways are actually composed of. To some extent in the brain, although I have to confess that the brain I find to be a big, complicated, confusing organ, and so I pretty much skip it and just go down. I'll maybe give people some questionnaires, but that's about the most I'm capable. We'll leave it to the real psychologists to help us with that. But in any case, what we're really trying to figure out is which if you start with the brain, which particular transcription factors, if you will, come to represent social information? In other words, there should be some binding between what's going on outside us and the activity of particular transcription factors, although, not much is known about that. We'd also like to know something about how these social signal transduction pathways interact with the human genome, or primate genomes for that matter, and to what extent polymorphism or individual variation in the genetic sequence of an individual might affect their sensitivity to this kind of social signal transduction. In the recent past, we've also started asking more questions about which particular cells are listening to social information, which cells are sensitive. Am I really right that your liver ignores your social world, whereas apparently your leukocytes are paying attention? I don't know, because nobody's giving me livers yet, but the general question of which cells are sensitive to this kind of dynamic and which aren't has turned out to be surprisingly illuminating as we go to try and theorize about why these kinds of connections might have evolved in the first place. So I'll take you on a hopefully pretty quick tour through some sort of representative examples of the kinds of answers we're getting from asking these questions. For instance, at the level of the genome, which genes care about your social world? Which genes seem to be sensitive to these kinds of dynamics? Fairly consistently, we were quite surprised. Anytime I run a study and I see anything close to replication, I am shocked to the point of just about, sort of paralysis. But it has been surprising the extent to which this particular study of lonely older adults versus integrated older adults have kind of recurred or reappeared in various subsequent studies. So I'm going to use this as just sort of a template for representing the kinds of patterns that we've now seen across a variety of different kinds of social or psychological adversities. One of the things we see fairly reliable, and the other caveat I'll give you is unless clearly stated otherwise, I'm mostly going to be talking about leukocytes because we can get a lot of those from lots of different people and monkeys. I'll occasionally talk about brains, but for the moment just realize that this is fundamentally a story, if you will, an existence proof for the proposition that social world can regulate gene expression that comes predominantly from cells of the immune system. So one group of genes that we see fairly consistently upregulated in individuals confronting adversity are genes that are involved in inflammation in the earliest stages of an immune response. Genes that are activated basically when cells detect that a cell near them has died or is injured or somehow similarly sort of decompensating. We also see changes in the expression of genes involved in cell growth and differentiation, and genes that are involved in transcription control itself. In other words, one of the things that we'll see fairly recurrently is changes in the expression of, for instance, the receptors that serve as the eyes and ears of a cell in terms of transducing information from the environment via its biochemical representations within the body. So fairly recurrently, we'll see upregulated inflammation, indications of cell growth and differentiation being regulated, and changes, if you will, in the infrastructure that maps inputs outside the cell into outputs in terms of gene expression. In downregulated genes, in cells of the immune system, we often see suppression of a couple of specific components of the adaptive immune response, the more sophisticated immune response that recognizes specific pathogens and tries to develop, if you will, sort of targeted immunological responses to them. We don't see general, if you will, sort of broad immunosuppression. We don't see the whole immune system just shutting down. In fact, some components, such as inflammation, are actively promoted. Lots of things seem to be left largely stable and intact, and a few components, such as the expression of genes involved in the production of a certain type of antibody, and quite recurrently, genes involved in what are called innate antiviral responses are suppressed. These innate antiviral responses basically represent the cell's first effort to trip up a virus if it should be infected. That's not the last thing the immune system is going to do. It's the very earliest stage of a response. But nonetheless, this seems to be a particularly recurrent theme across lots of studies. This suppression of this antiviral defense at the same time as this more pro-inflammatory response, presumably to extracellular pathogens, is being upregulated. Another set of sort of recurring themes is when we look in organ systems that have some degree of innervation. There's nerve fibers there. We often find that the genes that support those nerve fibers are upregulated in the context of adversity. So these are data from a study where John Capitano is looking at socially stressed rhesus macaques. What he does basically is houses them individually and then socializes them a couple hours a day with other macaques, either the same macaques every day, in which case they develop great macaque friendships and a stable social hierarchy and everything is fine, or they socialize with a different set of macaques every day. It's not profoundly stressful, but just enough to kind of make you feel like, well, I don't really know what's going on. You can think of it as a decent macaque model for the kind of subtle threat, or not even real threat, but sort of more uncertainty that you might argue pervades most of modern American life. And what he sees is about a twofold increase in the expression of, for example, this key gene here, nerve growth factor, which has the effect of essentially supporting the arborization of the sympathetic nerve fibers that are present in the lymph nodes, which are what we're actually looking at in this study. And I'll talk more, I think, a little bit later about what lymph nodes are, why they're important, and why we should care about this. But the key point for now is that genes that support, if you will, the neural pipeline that connects the brain and the social world to our tissues also seem to be subject to this kind of dynamic. In other words, this is part of that transcription control theme I was talking about earlier. As a function of that, you can see from that scatter plot over there, you get more nerve fibers in the lymph node. In other words, you get gene activation promoting the development of a bigger pipeline from the brain to the peripheral tissues. And then, of course, in the rat study that I described earlier, we've actually looked at a number of different brain sections, and we find similarly scaled changes in gene expression, very large numbers of genes that are up and down regulated in the central nervous system as a function of social environments. So those are some of the kinds of themes that recur on a fairly regular basis in terms of which genes are sensitive to these dynamics. Now remember, we have about 20 to 22,000 genes in our genome. I've been talking to you about something on the order of about 1,500 to, at the low end, maybe about 100 to 200 genes that are differentially expressed as a function of these kinds of adverse social conditions. So I don't want you to walk away from here feeling like, yeah, your body's undergoing wholesale crazy remodeling as a function of social adversity. There's lots of genes that are changing activity, but still, nonetheless, they comprise about 2 to, on the high end, maybe 10% of the genes that are actually present in the genome. So there's fairly selective targeting of these kinds of influences. All right, how does that selective targeting take place? In other words, what peripheral neurobiological systems and cellular signal transduction systems and transcription factors are actually carrying social information? We've got about, depending on your particular definition, between about 800 and 2,000 different transcription factors working in our cells. Which of those is, carrying information about the social world? Well, one way it turns out transcription factor work, while endlessly entertaining to us molecular biologists, is actually fairly difficult and tissue intensive. So we can't directly measure the transcription factors very efficiently in studies of human beings. So what we do instead is basically a bunch of bioinformatics tricks to try and get a guess about what's going on, and then we can go in there and do essentially sort of targeted confirmatory assays on maybe 2 to 5, sort of bioinformatically defined suspects, instead of having to go through all 800 to 2,000 transcription factors and laboriously test each one of those. We can do that with RNA really well. We can't do that with transcription factors very well. So we need some kind of first pass targeting analysis. And the way we do that is we take what we can do really well, which is measure RNA, and what we have done, although it was a little bit laborious, and that is sequence the whole human genome. We can take those two pieces of information and kind of cross them to make a guess about which transcription factors are involved. And the reason we can do that is that, one, we can measure all of the genes that are expressed in the human genome. Two, we have the human genome sequence, and we increasingly can basically sweep through what's called the regulatory region of these genes, the section upstream of where the so-called open reading frame or the transcribed or the, what did I call it earlier? Anyway, the structural region of the gene. And look for the DNA motifs that these transcription factors like to bind to. So it turns out that transcription factors can't bind wherever they want to. They need to, in some sense, see a particular stereotyped sequence of nucleotides. There's some flexibility in some positions, but they need to see some fairly reasonably conserved patterns, or else they can't bind to that DNA and they can't flag this particular gene for expression. So as we learn more and more about the specificity of transcription factors in terms of DNA, that allows us to go through the so-called promoters of these genes and ask, do we see any regularities in the nature of the DNA sequences that are upstream of the coding regions of these genes? From that, if we can make a guess about which transcription factor is actually driving the observed changes in gene expression, that allows us to make guesses about the upstream receptor systems that are activated, because we know a certain amount about how receptors couple to particular transcription factors. And then from that, we can make inferences about, if you will, sort of the biochemistry that's carrying this information. So another kind of cartoon representation is, if we have this directed flow of information where some kind of neural representation or hormonal representation of the social world ends up interacting with receptors, and that activates these transcription factors, they bind to the regulatory region of the gene and they turn on the gene like that. That directed flow of information, in principle, we should be able to reverse. Particularly if we can measure this section really well, and we can't measure the rest of this anywhere near as efficiently, it would be great if we could simply just reverse this flow of information and make guesses about this kind of stuff up here from the two things down there that we actually know a fair amount about. Now, that's a little bit complicated by the fact that most genes are regulated by multiple transcription factors, anywhere between 3 and maybe at the high end 12, 15 different transcription factors generally kind of conspire to regulate the activity of most of our genes. So we can't run that reverse inference program very well from one gene. In other words, we'll get to this point of the promoter sequence and say, well, we've got a bunch of candidates about what could be carrying the environmental information into gene expression. But on the bright side, we've got about maybe 10 to 100 times as many genes as we have candidate transcription factors. So we should be able to go across large patterns of gene expression and ask, what is different generally in the promoters of genes that are upregulated in this context relative to genes that are not upregulated? So if we go through here and we compare the genes that are upregulated in this cartoon, the ones that are marked in green in the coding region, with the promoter sequences of the genes that aren't regulated, what we notice in this particular version is, wow, that red cross transcription factor, that seems to be present in lots of cases where the gene's upregulated and generally not present in cases where the gene's not regulated. So from that, I can make a guess that maybe this red cross transcription factor is actually carrying this environmental information into changes in gene expression. And after that, I can make guesses about which receptors and neural or endocrine pathways might be involved. Okay. So if we pursue that agenda, for example, with these lonely older adults, what do we find? Well, we find that one particular family of transcription factors, the so-called NF-kappa B rel factors, seems to be particularly prominent in the promoters of the genes that are upregulated. And the promoters of the genes that are downregulated actually generally tend to have what are called glucocorticoid response elements in their promoters. These are the nucleotide motifs that the glucocorticoid receptor would bind to if it is activated. In general, we would expect that if there were a lot of hypothalamic pituitary adrenal axis activity, that we would find the genes that are upregulated have lots of these glucocorticoid response elements. Instead, we find actually, in general, it's the genes that are downregulated that have these things. So this gives us a hint that whatever is coming in through the brain and getting down into the genome here is probably not being carried by the hypothalamic pituitary adrenal axis. It's probably being carried through some other pathway because otherwise we'd see presumably upregulation of these glucocorticoid responsive genes. Now these aren't the only transcription factors that we find sort of indicated dynamics for across lots of different studies of adversity. But what's interesting about these two is that these have a kind of a fundamental yin-yang relationship with one another. And they actually control, if you will, the set point for inflammation, both under basal conditions and in terms of cells' responses to tissue damage. So NF-kappa B is the chief positive driver of inflammatory gene expression. And glucocorticoid receptor is supposed to be in the business of counteracting that dynamic. It's the physiologic brake for most of inflammation. So the fact that we're seeing this kind of reciprocal sort of upregulation of one and downregulation of the other, indicated by these kinds of promoter bioinformatics, actually turned out to be a pretty heuristically valuable observation. It gave us a lot of insight about where we should look for the, if you will, sort of the cellular or the molecular etiology of these dynamics. There are several other transcription factors that seem to be downregulated in terms of activity, particularly those that control the expression of interferon response genes. Not a surprise, since these type 1 interferon genes were among those that were particularly repressed. And then we see lots of transcription factors that seem to be playing an affirmative role in orchestrating the transcriptional upregulation, including a number of genes that we know from previous studies are, if you will, the handmaidens of the receptors that convey neurotransmitter signals, particularly the CREB-ATF family of transcription factors, very important in conveying neurotransmitter receptor signals. EGR, NRF, and GATA factors also play a major role in structuring these dynamics as well. Okay, so what I've done here is given you kind of a quick tour through how we can go from genome scale information to make inferences about these social signal transduction pathways. We can follow these up with more direct assays of you know, is this particular transcription factor activated? That's actually gone fairly well. That kind of bioinformatic analysis, if you can get enough differentially expressed genes, has been fairly reliable in telling us things that subsequently validate using other methods. So broadly speaking, this has begun to develop, if you will, sort of a dictionary about which specific transcription factors are hooked up to socio-environmental conditions as transduced through our brains and perceptions of the world. What can we do with that kind of information? Well, one of the things we can do is start to think about in a sort of a more sort of, if you will, forward or logic or model-driven way. Okay, if I know how social adversity turns into transcription factor activity, and I've got a sequenced human genome, I should be able to go out there and make predictions about which genes are sensitive to social regulation, even without any empirical information, you know, at hand about whether they actually are up or down regulated. In other words, I should be able to model this system going forward. And in fact, you know, if I do that, I should also be able to make guesses about which individual polymorphisms in those genes might make a difference in terms of both which genes get activated and not, but also which genes get activated in which people. In other words, if people differ in their genetic sequences, in the sequences particularly of those promoter regions, why don't I just show you the cartoon here. So if this is a scenario where the social world basically becomes an input and gene expression becomes sort of an output from the cell as mediated by, if you will, sort of the fundamental programming of the primate genome, is there a place where individual differences in DNA sequence can interfere with this, either sensitize or desensitize an individual's gene transcriptional response to socio-environmental conditions? Well, one obvious place of interaction is at this site of contact between the regulatory region of the gene and the transcription factor itself. So remember, the transcription factors need to see these stereotyped sequences of nucleotides in order to bind to the DNA and turn on, or sometimes they actively repress, the expression of a particular gene. So that creates the possibility that variations in the DNA sequence within that transcription factor binding site, if you will, have the potential to either increase or decrease the sensitivity of this dynamic. So if we can predict that, for instance, the GATA transcription factor, for example, is one biochemical representation of adverse social conditions, then we should be able to go through the human genome and ask, you know, one, which genes are targets for the GATA transcription factor, and then two, if we know something about polymorphic variation in the sequence of these genes, we should be able to make predictions about which genes respond differentially depending upon individual characteristics. So here is an example of how that worked successfully. Most of the times, this is not going to work so successfully. So, don't feel like, oh, they've got 20 more of these things up their sleeves or something like that. But this actually can be done on some occasions, and I'll tell you which particular occasions you can do it on at the end of this little interlude. But the basic idea is if we've got gene sequences that make it impossible for a transcription factor to bind, then that prevents that social environment from activating this particular gene. In other words, we'll still get the experience of social adversity. It will still be turned into biochemistry. It'll still activate transcription factors. You just won't get the same gene expression response from a cell in an individual with a suboptimal promoter polymorphism for that particular transcription factor. So here's an example of where this has actually worked reasonably well. So this is the human IL-6 gene, an unusually influential gene, and by the way, that's the answer for why this doesn't happen all the time and happens sometimes, is that most genes actually are not terrifically influential by themselves. Genes mostly do what they do as elements of larger conspiracies. IL-6 just happens to be an unusually influential gene because of its role in structuring inflammatory reactions and thereby fertilizing the development of lots of the diseases that kill us these days. So in the imaginary world of the computer, actually in the real world, we have the human genome sequence. We can then go and computationally take the human genome sequence, search for places where these, if you will, socio-environmentally responsive transcription factors, like the GATA1 transcription factor, bind to. And in the human IL-6 gene, it turns out about 174 base pairs upstream of the transcription start site for that gene is this sequence right here, which the GATA family of transcription factors loves to bind to. So this is a number that goes on a 0 to 1 metric, and you can think of it as sort of approximating something like a correlation coefficient in terms of, except it actually goes only from 0 to 1. Forget the correlation coefficient analogy there. Bad plan. 0 to 1, let's stick with that. We'll even quit the metric and just say 0.94 in general. In the real world, a transcription factor will bind to this particular nucleotide motif. About 20% of people of Northern European ancestry have at this site in their IL-6 promoter, not this G right here, but a C. That's the only switch, but changing that G to a C gives you a DNA motif that the GATA family of transcription factors generally won't bind to. Just flipping out that one nucleotide alone pretty much destroys the consensus sequence that this transcription factor wants to bind to. So in my own little imaginary world of computer programming, this should make the IL-6 gene essentially less sensitive to any information that the GATA family of transcription factors carries, including what we had already determined, as I described earlier, the role of social environments, and adverse social environments in particular, in activating that transcription factor. So great, nice computer trick. Does that work in the world? Well, my version of the world is you get some blood cells and you put them in a test tube. So let's just try that world, which is a little bit tractable, and take we'll call it a biochemical representation of adversity in the form of sympathetic nervous system activation. So norepinephrine is the major neurotransmitter released by sympathetic nervous system neurons during fight or flight stress responses. So I can take some white blood cells, I can engineer them so that we can measure really efficiently and accurately the amount of IL-6 gene expression. And then I'll dose those cells with norepinephrine, my sort of model of physiologic stress response. And you can see that under normal circumstances, that is with the ancestral or wild type version of the IL-6 promoter, you get about maybe an 8 to a 10-fold increase in the amount of IL-6 gene expression. If you switch that G to a C and you carry out that same experiment, you get much less response. So maybe about a 2 to a 3-fold increase in IL-6 gene expression. So not nothing, but a big change in, if you will, the intrinsic sensitivity of this gene to this, admittedly sledgehammer laboratory representation of what presumably is a, likely less intense, more subtle dynamic under most circumstances in the real world. So yeah, this promoter polymorphism does make a difference to the cell's intrinsic capacity to respond to this particular transcription factor. So, that would not be a bad kind of cellular and genetic trick, but still we'd like to know, does this make a difference for human health at all? So we were lucky enough to have access to a study of older adults in basically the east coast of the United States, which captured those individuals when they were 70 years old, recruited them when they were, as healthy as you can be when you're 70. Actually, in general, they were doing great. And so, this is not a study of people who are already sick or getting ready to die. But what they did in the MacArthur study of healthy aging is follow those individuals. And as you can see, we were able to follow them, they were able to follow them for another 20 years or so, which is through the frame of most mortality in the United States at this point. So if that story I told you a moment ago is really true and matters in the real world, then what we should see is that people who are exposed to substantial adversity in their life should show essentially increased incidence and, in this particular scenario, mortality due to inflammatory causes of death. In other words, the things that IL-6 feeds should operate with more intensity for individuals who are confronting substantial adverse circumstances in their life. That should be true for individuals, the vast majority of us, who are carrying the so-called ancestral or G allele of the IL-6 promoter. And that's actually exactly what we saw. Again, much to my surprise, anything that works out generally surprises me, especially if we're jumping from leukocytes in cell culture to epidemiologic mortality data. But in this particular case, what we were doing is looking at measures of depressive symptoms as an indicator of substantial adverse life circumstances. And we had done, the important, obvious checks of making sure that it wasn't the IL-6 genotype itself that was causing depression. That's independent of these kinds of dynamics, no substantial association there. The reason looking at depressive symptoms made some good sense is that most of the variability in whether you're depressed or not at any given point in time has to do with the circumstances in your life at any given point in time. There's a little bit of genetic influence in how vulnerable you may be to adverse life circumstances, although, frankly, we're hard-pressed to point to where that actually is, and it's certainly not in the IL-6 gene. But to make a long story short, most of whether you have depressive symptoms has to do with whether you feel like life has beaten you down, essentially, or deprived you of somebody or something that you really care about and value deeply. So we took this as a sort of a reasonable, if you will, sort of rolled up general proxy for substantial adversity, no matter how its particulars came about. What we're really interested in is it experienced as something severe and overwhelming. So what you can see here in these survival curves is that for individuals who have essentially the sort of the goddess-sensitive version of the IL-6 gene, there is this accelerated mortality curve relative to individuals who are non-depressed, that is presumably not confronting major, overwhelming, severe life adversity. And that is quite a substantial acceleration. It's, I think, something on the order of a 3 to 4 year acceleration in average mortality times. That's all true for people whose IL-6 gene is hooked up to GATA transcription factors as representations of adversity. For people who do not have a GATA-sensitive IL-6 gene, there's absolutely no acceleration in mortality. Now, the reason this works for us is that 70 to 90-year-old individuals die in the United States predominantly of inflammation-mediated causes of death. Not exclusively. There's a substantial fraction of disease sources that are not not inflammatory in etiology. But this is a major driver of contemporary human mortality in developed nations. So that made a certain amount of sense. We went through and tried out a couple of other things to try and convince ourselves this is really true. We've got good biomarkers of IL-6 activity. So one thing we can do is just say, if we control away all of the variance in mortality that's due to variations in IL-6 activity as marked by C-reactive protein levels, we can, in fact, knock out that association entirely. In other words, if we look at people who, well, I'm not going to drag us through the weeds on that in the interest of time here. We can also look at non-inflammatory causes of death and ask whether we see the same genetic relationships there. We don't see anything of the sort. So for inflammatory causes of death, things like atherosclerosis, many types of cancer, neurodegenerative diseases, this relationships hold. For things like accidental death, infectious mortality, that kind of thing, we don't see any substantial contribution from these dynamics. So what's happening here basically is we've got a system where adversity becomes represented as sympathetic nervous system activity. It interacts with, basically releases neurotransmitters that interact with particular receptors on the surface of the cell. That activates a number of transcription factors, including the GATA family of transcription factors. GATA factors in particular can interact with the IL-6 gene and promote the expression of the IL-6 gene, which then fertilizes inflammation and essentially, essentially enhances mortality risk. So this whole input-output relationship between adversity and mortality risk can be abrogated if people have this polymorphism in the IL-6 gene that prevents this biochemical representation of adversity from actually playing on the IL-6 gene. In other words, you don't get the output in terms of mortality from that system. Now, the story I just told you about genetic polymorphism, I think this is certainly not the only way that these kinds of dynamics can take place. You can have, for instance, epigenetic polymorphism in some sense, not technical polymorphism in the sense of variations in DNA, but you can definitely have, for instance, the promoter region of the IL-6 gene or any other gene methylated, for example, in a way that prevents transcription factors from binding to the promoter and producing the gene transcriptional output that they would have otherwise. So there's lots of, well, I won't say necessarily lots, but there's a reasonable number of sort of, if you will, intervening variables that can disconnect, if you will, the basic input-output relationship between these kinds of socio-environmental conditions and gene expression, both genetic and environmentally modifiable. Let's see, I think in the interest of time, I'm going to skip that one for a moment. Sorry for that, because I want to get to these stories about which particular cells seem to be sensitive to these dynamics. So in addition to asking which RNAs track which transcription factors, which is implicitly what I was doing earlier, we could also ask which RNAs track which particular cells. The definition of a cell, to some extent, has to do with which genes are actively expressed. That's what makes a liver cell, a neuron, a neuron. They have the same DNA. What differentiates them is which subset of genes are actively being expressed. So, if that's true, we should be able to go through the list of genes that are essentially differentially expressed in any given general tissue environment and ask, are they predominantly from one type of cell or another? So as we've done this in the context of white blood cells, which it turns out are a gamish of about, depending on your level of specificity, about 12 to maybe 40 different types of cell, what we find fairly recurrently is that most of the genes that are differentially expressed are being contributed by two types of cells that are involved in the earliest, if you will, decision-making and first line of defense response to tissue damage, monocytes and dendritic cells. The smarter, more sophisticated, and sort of later cells in most immune responses generally aren't contributing substantially to the total white blood cell shift in gene expression. The reason that that was a helpful observation is that it made us ask or think more deeply, I guess, about what, if you will, I apologize for this personification of cells, but what is the immune system trying to do when it's turning these genes on and turning these genes off? In its job as defender against pathogens, why are we seeing this, for instance, upregulation of inflammatory genes and growth and differentiation genes and these downregulation of genes involved in antiviral responses in particular? These are actually Steve Sumi's rhesus macaques that have been reared either with their mother in standard sort of rhesus macaque social conditions or peer reared. That is, their mom is not around. They're being reared in incubators. They're now about 3 to 4 months old. Within the first 3 months of a macaque's life, you can already see this conserved transcriptional response to adversity, this gene expression program coming on, and that will presumably shape the way their cells respond to injury and tissue damage and disease as the rest of their life unfolds. So anyway, why is it that we're getting this shift basically? Well, essentially what's happening is these particular cells are essentially changing the extent to which they are, under basal conditions, deploying their gene expression resources toward viruses, essentially, which is what they're doing well under the mother-reared condition. What they're basically doing is they're clearing out some space in their transcriptome to upregulate the expression of genes that would help defend the organism against bacterial infections and other kinds of extracellular pathogens. These turn out to be two fundamentally different types of immune response, and these guys have learned how to feed on the genes that are expressed to defend us against those guys. So humans and microbes have been, oscillating back and forth in an evolutionary arms race long before we were humans, probably, so that these two types of immune response, a good immune response against bacteria, that is a highly pro-inflammatory, antibody-rich immune response, is to some extent incompatible with a great response against intracellular pathogens like viruses, that is a strong type 1 interferon response. So what we're seeing is downregulation of this great type 1 interferon-mediated antiviral response, upregulation of the kind of response that protects you against extracellular pathogens. Okay, well, at a microbial level, that makes a certain amount of sense, but why would you do that? Why would you hook up this kind of decision-making to the social world or our experience of our environment as being, sort of promising and supportive versus threatening and uncertain? Well, the answer is that the social world has a lot to do with our microbial ecology. In fact, you guys are probably better positioned than anybody on campus to think about this, just because the way we live now is profoundly different than the way we lived over the majority of our evolution, and the implications of that for infectious diseases are particularly dramatic. Viruses need us to infect one another. Viruses are fragile and highly species dependent. If you get a viral infection, it has come almost certainly from another member of your species that you've spent a fair amount of close physical contact with. That is not true for bacteria. Bacteria are hardy little creatures. They live on surfaces. They live on the teeth of predators. They live on us all the time. They live in us. And bacteria basically have a mode of transmission that is, if not totally social, at least only incidentally social. They can get us basically no matter what happens. Not true for viruses. And so most of the anthropological speculation and analysis now suggests that major viral infections are relatively recent developments, probably within the last 10 to 100,000 years. We've become, sort of much more subject to these kinds of, if you will, close social contact mediated disease transmission processes, just because we didn't live in huge groups much before that. And we were pretty mobile and a number of other things that turn out to make a big difference for infectious disease propagation. So what's happening here is it appears that the immune system is changing the basal sort of defensive stance that it has away from a fundamentally antiviral stance and toward a more, if you will, antibacterial stance when individuals move from, if you will, sort of favorable or salutary social conditions to more threatening or hostile social conditions. So again, the reason that makes sense is that close social contact is a superhighway for viruses. That has changed fundamentally. Human sociality and our ancestor sociality has changed fundamentally the epidemiology of the microbes that we confront. So if close social contact basically is a superhighway for viruses, isolation and hostility is a superhighway for bacteria. Why? Well, what happens when your conspecifics don't like you? They smite you. They injure you. They basically make it much easier for the bacteria that's living in your gut or on your skin to go into your tissues and actually colonize the places that they shouldn't be and cause us disease. Same thing happens if we get banished from our social groups. A lone human being, as you guys understand better than anybody else, is somebody else's lunch. We're not big, we're not fast, we're not strong, we're not covered with a, sort of a thick, unbiteable carapace. We basically succeed or fail based on sociality, clever large masses or meta-organisms, that kind of thing. So to the extent that bacteria are coming in either through wound-mediated injuries from conspecific hostility or coming in through sort of the kinds of wounding that is associated with predation of us when we're loners, you have the beginning of, if you will, enough stochastic architecture for an immune system to eventually, over time, evolve, if you will, the heuristic that if you're with people and they like you, defend yourself against viruses. If you're on your own or with people that don't like you, don't worry about the viruses, because nobody is really going to be spending a lot of time, sort of breathing with you all day long or mating or anything like that. On the other hand, your risk of injury is much, much higher. So that led us to speculate that there might exist within the human genome at least two different modes of regulatory organization hooking up gene expression to the external social environment. A so-called integrated mode that I think is probably the default for human beings, and a sort of an isolated or an autonomous mode. In the integrated mode, we're, by virtue of our integration into close social networks, exposed to a large number of socially transmitted pathogens, especially to viruses. And so in that context, it makes sense to, by default, throw up a pretty good, sort of, if you will, hedged bet that viruses are going to be your major threat and upregulate the expression of type 1 interferons and immunoglobulin G. On the other hand, if you are isolated, then it makes more sense to don't worry so much about the viruses. They might get you someday, but the bacteria will get you faster. So you've got no social support, no social resources. You need to defend yourself more efficiently against the bacteria associated with wounding injury. So you upregulate pro-inflammatory cytokines and the production of other antibiotic isotypes that are good at protecting you against those kinds of extracellular pathogens. Now, in theory, that made pretty good sense historically, because there was this pretty good mapping between our experience of the social world as being there and supportive versus either not there or not supportive, and the nature of the kinds of pathogens we were exposed to. In other words, that gave us the capacity to use our brain to predict the future. The key to this sort of allostatic theory of physiology, that the reason we are such terrific physiologic creatures is because we can, in some sense, anticipate shocks and buffer ourselves against them, as opposed to, in classic homeostatic theory, just sitting around and waiting for a shock to hit us and then kind of trying to, respond after the fact. So this makes pretty good sense to hook up your immune response to your forecasts about the social world and its implications for microbes. But, as these stories always go, this plan is not serving us so well right now, because what have we done? Well, we spend our lives threatened and uncertain these days. So we're constantly basically activating autonomous mode defenses. We're upregulating pro-inflammatory cytokines, upregulating other components of the immune system at the expense of type 1 interferons and other elements of antiviral defense. So in a world where we've got antibiotics and we really don't need to worry about those pathogens so much, we're still running a body that is good against bacteria and also good at promoting atherosclerosis, neurodegenerative diseases, and cancer metastasis. And we're running a body that is, not really doing a terrifically good job of defending us against viral infections. It's certainly not completely out of the race, but it is definitely diminished in efficiency. So that's kind of a little bit about the agenda that we can extract from reasoning about cells and what they're trying to do. Another kind of dynamic involves the cells of the immune system, the nervous system, and why they would be upregulating the way they are. Why is it that we'd have, as part of our program, this upregulation of, for instance, nerve growth factor by social adversity? Well, this is what the lymph node of a rhesus macaque reared under stable social conditions look like. Each green dot there is basically a point source of norepinephrine. This is where the sympathetic nervous system delivers a signal to immune cells. This is a similar lymph node from a rhesus macaque reared under unstable social conditions, or actually just housed. This is only, I think, maybe about a 9-month-long experiment. But it turns out within even just a couple of weeks of unstable social conditions, these animals upregulate the expression of nerve growth factor that causes their neurons to arborize, and you have more point sources of catecholamines, a bigger pipeline from the brain to the immune system. The lymph node, it turns out, is the convention center for the immune system. That's where one type of cell gets together with another type of cell to coordinate their responses to tissue injury someplace else in the body. So if you're doubling the number of catecholamine point sources on the average, any response that the sympathetic nervous system is regulating is going to happen with greater intensity in these animals that have been subject to these unstable social conditions. So in these rhesus macaques, for example, you see about an 80% reduction in the amount of type 1 interferon that these things are basically making under basal conditions. So remember, I talked about this suppression of the antiviral response. That happens in spades for these rhesus macaques. And we've seen it across a number of other animals as well. We see it in Steve Suomi's animal models. Fairly reliable finding, and not a good one from an infectious disease standpoint. So if John Capitano infects these monkeys with SIV, the monkey version of HIV, they'll get sick much worse and much faster because they are not able to mount an efficient type 1 interferon response to that viral infection. So one of the things that is significant about that is if we can upregulate, in this case what's happening is nerve growth factor is getting expressed, and it is upregulating this, what I'll call peripheral distribution biology, these pipelines from the brain to the rest of the body. That creates this sort of recursive persistence. In other words, to the extent that I've got now more nerve fibers bringing this information down, that's just going to propagate more nerve growth factor expression. So this kind of dynamic becomes an opportunity for transient experiences or transient social conditions or circumstances or environments to change your body in a way that may perpetuate over time, either by leveraging future experiences of adversity and magnifying them, or by, and to some extent, the positive feedback loops that happen within the genome, where once you turn a gene on, sometimes it will just keep, with positive feedback, promoting its own expression. So that can happen at every step of the system. That happens probably at the level of the brain, as for example we were seeing in those rats that were reared in isolation. And it looks like it can persist for a fairly long time. So in one of my sort of favorite studies that I was lucky enough to write along on, but the idea really came from Greg Miller and Edith Chen. They thought, what would happen if we took a bunch of people who are enjoying good, solid, supportive social environments right now and just ask whether we see any differences in gene expression as a function of the social environments that they were born into and spent the first few years of their lives in. In other words, can we see any fingerprint of your early life social experiences, either adverse or favorable, on your gene expression profile right now, independent of what's actually happening right now? Now, unsurprisingly, what's happening right now contributes the majority of the variance. But they were still able to see changes in gene expression along this sort of more pro-inflammatory dimension that appeared to relate predominantly to what was going on early in your life. This was happening several decades later in these people's adulthood. So it is at least in principle possible. There's a, if you will, sort of an affirming the consequent kind of feel to this thing, but nonetheless, it's in principle possible that substantial periods of adversity, particularly during early development, may well persist at this transcriptional level over decades later into adulthood. So that creates this notion that somehow your genome may be, if you will, developing a theory about what the world is like. So this notion of defensive programming that Michael Meany has talked about, that then shapes how you're processing incoming experiences and may also sort of perpetuate itself at the molecular level. So you can kind of think about bodies in some sense as machines for turning environments into outputs of various sort. Those outputs can be behavior. They can also be gene expression. The reason the gene expression is particularly worth noting is that that then shapes the nature of your body in the future. In other words, if you've spent time 1 in an adverse environment, the body you have at time 2 will be, at least at the subtle molecular level, somewhat different. That raises the prospect that a certain environmental exposure at time 2 will yield different responses than it would have had you had a different history of environmental exposure. In other words, you'll get different behaviors, you'll get different gene expression profiles out, and so forth, propagating over time. So in that sense, this if you will, trajectory of gene expression from time 1 to time 2 to time 3 becomes sort of like a molecular biological record of your body's adaptation to the various environments that it's confronted over its individual history, in the same way that we think about the human genome at the DNA level as being that kind of a record of our response to the environments that our species has encountered. So the take-home point I want to leave you with is basically that your social environment today will influence the molecular characteristics of your body for the next 2 to 3 months. That is, sort of, I think, reasonably empirically defensible. It's not going to, cause you to turn into a blob of jello or a completely different organism, but it is subtly remodeling the profile of genes that are being expressed in your cells. Not most of them, probably about 2 to 10% of them. But these 2 to 10%, at least in the context of the immune system, are unusually influential. They promote inflammation, they suppress antiviral responses, they make animals get sick when they wouldn't have gotten sick, they seem to promote early mortality. And that's just in the immune system. We haven't even looked in the brain, we haven't looked in the liver. Maybe the liver will prove me wrong and turn out to be much more socially responsive than I gave it credit for. But in any case, the take-home point is that the social world gets into our bodies to a surprising extent. It gets deeply into our bodies. We can find ovarian cancer cells that are running hundreds of genes differently than they would have if this individual had great social support as opposed to the poor social support that they have. So all kinds of tissues seem to, one way or another, be getting messages from the social world. So on some level, that is pretty terrifying, right? I mean, let's go back to the beginning. I'm me, I'm the same me today as I was before, and I'm the same me if I'm in a different environment as I am in this environment. Well, not true at the strict molecular level. So on some level, as a, sort of a good Western individualist, that does not make me happy, right? I mean, this notion that where I'm going gets inside me and stays with me and becomes part of my sort of molecular developmental history, not acceptable. But on the other hand, it turns out, as I'm constantly trying to give myself reasons not to kill myself and despair of my children's lives, I figure, actually this is a great opportunity. So yeah, it's true, the social environments that you choose, the social environments you inhabit make a big difference, but that does give you the capacity to, at least indirectly, regulate these biological dynamics by, essentially the power of will and choice. So to the extent that we do choose environments that are most consonant with us, there's a decent chance that we'll be running our own personal genomes in the ways that are best for us. So that does, I think, become the only sort of hint of redemption in this whole system as far as I can tell. And I should, of course, conclude by thanking the people who made all this work possible, the people who actually collected data. At this point, it's been a long time since I collected any primary data from humans, for good reasons, because they kept not showing up for experiments and causing all kinds of trouble. But nonetheless, the people who do put up with that stuff, wonderfully, are Greg Miller and Edith Chen, John Cacioppo, Susan Lutgendorf and Anil Sood, John Sheridan and Nicole Powell, whose work really kind of helped us understand that dynamic with reduced glucocorticoid signaling and increased pro-inflammatory gene expression. John Capitano and Erica Sloan with the UC Davis studies of the virally infected monkeys, Margaret Kemeny, Jerry Zak, Teresa Seaman, Andy Follini, and Steve Sumi, Jim Heckman have also done great work with the primate models, particularly in the early development. So that, I believe, is all I have to say. I'm running a meager 20 minutes over time here. I apologize for, sort of dragging you guys too much farther into the afternoon here, but thanks for your patience.